One of the big unanswered questions about MS is what causes it? Well, it does seem to me like they're closing in on a very strong possibility. Hello, and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with MS for over 40 years. We have been talking a lot about possible causes of MS. I've done several videos about the intriguing possibility that Epstein-Barr virus causes MS, or at least plays an incredibly important causative role. Even though almost everyone in the population has been exposed to it and only a few of us get MS, still they're thinking there's a viral origin to the disease. Today I want to share with you some information about another virus that may very well be implicated in MS, and that is the herpes virus. This article appeared in Multiple Sclerosis News Today on July 21st of 2025. It's entitled Cold Sore Virus May Play Role in MS, Mouse Study Finds. Herpes simplex virus found to degrade myelin sheath. A common virus that causes cold sores, herpes simplex virus type 1 or HSV1, may contribute to multiple sclerosis. That's according to a study by researchers at the University of Illinois, Chicago. In mice lacking a protein that helps control HSV1, infection led to the degradation of the myelin sheath, the protective coating on nerve fibers that's damaged in MS, triggering MS-like symptoms. When those mice were treated with a molecule that blocks MLKL, a protein that boosts HSV1 infection, myelin was preserved. Our findings enhance our understanding of how viruses develop and offer potential avenues for mitigating viral-induced or nerve cell damage, Deepak Shukla, PhD, a professor at UIC, and the study's lead, said in a university news story. The study, MLKL-OPTN axis, regulates herpes virus-induced neurological sequelae, was published in Clinical and Translational Medicine. And I just want to point out here, since they have linked to it in the article, that back in November of 2023, they published an article called HHV6A herpes virus tied to more nerve damage before onset of MS. So they've actually had this in their sites for quite a while. And of course, he talked about other viruses linked to MS, because various folks have postulated that MS has a viral origin. And I'll put a link below of a video or two that I've done over the years on Epstein-Barr virus. Dr. Gavin Giovannoni and others have promoted that as a possible cause of multiple sclerosis. So we're seeing here that herpes, which is another virus, does seem to have ties to MS, and that really does seem to strengthen this idea of a viral origin. And they go on to say here that in MS, a mistaken immune attack on the myelin sheath also damages nerve cells and oligodendrocytes, the cells that produce myelin, giving rise to various neurological MS symptoms. One of the strongest risk factors for MS is a history of infection with the Epstein-Barr virus, a type of herpes virus which raises the risk of developing the autoimmune disorder by more than 30 times. Other herpes viruses, including the varicella-zoster virus that causes chickenpox and shingles, and human herpes virus 6, have also been linked to MS onset and progression. And by now you're thinking to yourself, do I get cold sores? And there are those of us who have gotten them and those of us who never have had them. I had them when I was younger, but I haven't had one in a long time. And when I do get them, it's usually stress-induced. What we do know about the herpes virus is that once it's in your system, it's in there. It just sometimes goes dormant or at least is asymptomatic, but it's always in there, and it can cause problems at any time. HSV1 causes oral herpes, commonly known as cold sores, and is carried by 2 of every 3 adults. While most people with HSV1 have no symptoms, the virus, which infects through the eyes and mouth, can stay in the body for life. It poses a particular risk to people with weakened immune systems. If you are infected, then your immune system is constantly locked in battle with the virus, Shukla said. And if for any reason you become immunocompromised, the virus can escape and damage your brain. In previous work, Shukla's team discovered a link between HSV1 and a heightened immune response in the brains of mice, leading to cognitive impairment, lack of coordination, and anxiety. I don't know, I've never seen a mouse with a cold sore, but then again, I've not really hung out with lab mice very much. So I guess in the lab they can do all kinds of things. The researchers also identified a protein, optineurin, OPTN, as an antiviral factor that restricts HSV1 replication and spread. Cells without optineurin were more susceptible to programmed cell death after HSV1 infection. Now the team found that a protein called MLKL boosts HSV1 infection in the absence of optineurin. Without optineurin, MLKL also facilitated the transport of the virus to the cell's nucleus, where it replicates. Further experiments confirmed that optineurin controls HSV1 infection by triggering the degradation of MLKL. To investigate whether MLKL influences HSV1 infection in animals, the researchers infected the eyes of mice with and without the gene for optineurin. In line with cell-based results, mice lacking the optineurin's protein exhibited higher infection rates 4 days post-infection, alongside elevated MLKL production. Imaging studies revealed extensive clumping or aggregation of MLKL in the brains of optineurin-deficient mice, but not in control mice, in response to HSV1 infection. Also, these clumps appeared to trigger the death of myelin-producing oligodendrocytes. Oligodendrocyte death and myelin loss occurred in brain regions that play essential roles in coordinating movements and maintaining balance. Well, there you go. That might be my problem right there. In behavioral studies, mice lacking optineurin showed significantly impaired motor coordination and shaking behavior after HSV1 infection. The team also found aggregates of MLKL in the spinal cords of a standard MS mouse model. There were also lower levels of optineurin protein in regions of MS lesions in brain tissue collected from human patients compared with healthy counterparts. However, when the team treated optineurin-deficient mice with necrosulfonamide, an MLKL blocker, myelin loss was prevented. In comparison, acyclovir, the standard of care for herpes infections, reduced viral loads but did not reverse demyelination. Very interesting. Okay, well now we see that there might be some good coming out of this for both people with MS and people with herpes. These data suggested that the intricate link between MLKL and OPTN that serves to maintain myelin, with dysregulation of this balance resulting in severe impairments in motor functions and behaviors reported typically in MS and MS-like demyelinating disorders, the researchers wrote. Treatment with an MLKL inhibitor demonstrated therapeutic potential by preserving the myelin sheath and reducing neurologic damage during HSV1 disease. I thought that perhaps it would be good to just put in this video too, because it does talk about herpes virus and Epstein-Barr virus as causative of MS. And I guess it's escaped my mind that Epstein-Barr virus is actually a form of the herpes virus. So that was interesting for me to remind myself of. So let's take a look at this video. I want to point out that I think this is an AI narrator because she continues to refer to MS as Ms. because that's what it looks like to the AI program, I guess.
Researchers at Karolinska Institute have developed a new method to separate between two types of a common herpes virus, HHV6, that has been linked to multiple sclerosis, MS. By analyzing antibodies in the blood against the most divergent proteins of herpes virus 6A and 6B, the researchers were able to show that MS patients carry the herpes virus 6A to a greater extent than healthy individuals. The findings, published in Frontiers in Immunology, point to a role for HHV6A in the development of MS. MS is an autoimmune disease that affects the central nervous system. The cause of the disease is unclear, but one plausible explanation is a virus tricks the immune system to attack the body's own tissue. Human herpes virus 6 (HHV6) has previously been associated with MS, but in those studies, it wasn't possible to distinguish between 6A and 6B. Through virus isolation from ill individuals, researchers have been able to show that HHV6B can cause mild conditions such as roseola in children, but it has been unclear if HHV6A is the cause of any disease. According to estimates, as many as 80% of all children are infected with the HHV6 virus before 2 years of age, and many also carry protection in the form of antibodies against this particular virus for the rest of their lives. But since it hasn't been possible to tell the two variants apart post-infection, it has been difficult to say whether HHV6A or B is a risk factor for MS. In this study, however, the researchers were able to distinguish between the A and B virus by analyzing antibodies in the blood against the proteins, immediate early protein 1A and 1B, i.e. 1A and i.e. 1B, that diverge the most between the two viruses. This is a big breakthrough for both the MS and herpes virus research, says Anna Fogdel-Hahn, associate professor at the Department of Clinical Neuroscience at Karolinska Institute and one of the study's senior authors. For one, it supports the theory that HHV6A could be a contributing factor to the development of MS. On top of that, we are now able, with this new method, to find out how common these two different types of HHV6 are, something we haven't been able to do previously. The researchers compared antibody levels in blood samples of some 8,700 megasiemens patients against more than 7,200 healthy people whose gender, date of birth, date of blood sample and other factors matched those with MS. They concluded that people with MS had a 55% higher risk of carrying antibodies against the HHV6A protein than the control group. In a subgroup of almost 500 people whose blood samples were drawn before the onset of the disease, the risk of developing MS in the future was more than doubled if they had a 6A viral infection. The younger the people were when the virus was first discovered in the blood, the higher the risk was of developing MS in the future. HHV6B, on the other hand, was not positively associated with MS. Instead, MS patients had lower levels of antibodies toward IE1B than those without MS. Antibodies toward Epstein-Barr virus (EBV), another herpes virus that is also associated with MS, were analyzed with the same method, and the researchers were able to show that individuals affected with both viruses had an even greater risk of MS. This indicates that several virus infections could be acting jointly to increase the risk of MS. Both HHV6A and 6B can infect our brain cells, but they do it in slightly different ways. Therefore, it is now interesting to go forward and attempt to map out exactly how the viruses could affect the onset of MS, says Anna Fogdel-Hahn.
I'd also point out that this video was posted in 2019, so they've understood this connection was there long before we read about it in the recent news article. Have you had exposure that you know of to either of these two viruses that you might likely be carrying the antibodies for them? That might be something you could even have your doctor test for. Of course, once we have MS, it doesn't really matter if we know what the cause is, do we? Because we have to live with it one way or the other. But it might be something for someone to think about who doesn't have MS, and maybe they could just have their doctor run a blood panel or something and see if they have those antibodies. But like I said, almost everyone in the population has Epstein-Barr virus antibodies, and I would say well over half the population has been exposed to the herpes virus as well, even though most people are asymptomatic. So I don't really know what good any of this does us in the long run, but it is interesting to know that there is very likely a viral origin for multiple sclerosis. So of all the unanswered questions, at least perhaps that's not one of them anymore. But that's all I have for you today. So until my next video, please do keep taking really good care of yourself, and I'll see you again soon.